This is joint work with the University of Pittsburgh and also Oak Ridge National Labs. Just a second. Okay, so the primary concern of our lab is to build methods that aid in the interpretation and protein dynamics. So on the left, hopefully, you see just a movie, and this is depicting a simulation of ubiquitin that we've run. These are downsampled snapshots from 500 nanosecond simulation. And more generally, we're interested in how we can decompose the landscape that is spanned by a simulation in a meaningful way. So go back. So on the right side, you see a depiction of an energy landscape. We're probably familiar with this, where small maxima and minima correspond to small fluctuations, thermal fluctuations, and the large substates, which are separated by hills and valleys, correspond to more substantial substates. And the transitions between substates might have functional importance. So that's what's depicted there. But along the x-axis, I say conformational coordinate, and this is just a schematic because it's not obvious what to pick for that axis that actually resolves the landscape or the substates in a meaningful way. So we're interested in building methods that enable a user to find a conformational coordinate that is meaningful for the function of the protein. And for ubiquitin, which we'll be talking about today, the substates are particularly important because ubiquitin can bind many, many different substrates as part of the degradation pathway. So there's a primary binding region for ubiquitin on the top, and the secondary binding site is on the right. So the basic question is, can we identify substates in a meaningful way such that we can perhaps explain what facilitates this binding? And if you're interested in modeling protein motions or even protein structure, like we heard from before, one thing that you're up against is just the range of time scales and motions that proteins undergo. So from femtoseconds up to seconds, of course. And there's always going to be a trade-off between the spatial detail that you can get versus the temporal sampling of what motions you're observing. So we're interested in trying to somehow bridge the detail and sampling that you see in both experimental or computational methods. So to introduce the aspect of my talk in the title that's called Quasi-anharmonic, to introduce anharmonicity, I'm going to show you a comparison between neutron scattering data and MD data. This is actually for a different protein, but in the solid, I'll use the mouse, in the solid red line here, this is the radius of gyration, the distribution of radius of gyration from neutron scattering data. And two simulations from MD, the same measurement was computed, and that's shown in solid blue and dashed red. And you see that in the neutron scattering data, there's a substate that just isn't accessed in the MD data. But you should also notice that if I did a good job of characterizing what happens in the tails of the MD data, at least I'm getting close. At least I'm getting closer to the substate that is actually present in nature, or at least in the solution. So this plot is an example of many that we've seen when analyzing dynamics data, where we think we should be looking at the tails of the distributions, and we should be looking at deviations, as in dynamics, which are anharmonic, which represent higher probability in being in the tails than elsewhere. So we've actually developed a particular method for going from one-dimensional sparse data to alpha carbon resolution models of proteins, and that's at the poster tomorrow. And I'll bring in some of these ideas with the rest of the talk. After pursuing these long-tail distributions, which I'll describe in more detail, we've come up with this analysis method called quasi-anharmonic analysis, which is trying to find, again, the most anharmonic motions within a molecular dynamics simulation. And I'm showing at the bottom some of the timescales that these methods are applicable for. And what I'll get to at the end of the talk is whether we can model protein dynamics very compactly in a linear fashion, but only after determining the substates using quasi-anharmonic analysis. And this is going to enable us to bridge a hopefully a larger section of this timescale spectrum, which a lot of us are probably often facing when trying to analyze protein dynamics. Or if you're using experimental methods, you know there's the same trade-off. So under, again, the overall heading of trying to very compactly model some model the dynamics both within substates and between them, we're going to have to first find some low-dimensional subspace, which is useful for comparing conformers from an MD simulation. We're going to do some clustering to try and identify substates, again, in an energetically meaningful way. And finally, I'll show the linear model we've come up with for easily modeling the dynamics within those subspaces. And just in general, we found that a lot of machine learning and inference techniques have been very useful for this problem because we're commonly dealing with data, whether it's dynamic, computational, or experimental, that's ill-posed or sparse in some sense. Okay, so I showed you the long-tail distribution in the experimental data, and now I want to characterize what I mean by global anharmonicity. What does it mean for a protein to be acting in this way, non-Gaussian way? And the left plot is actually showing the deviations, the histogram of deviations for, again, the ubiquitin simulation that I showed at the beginning. So the red line is showing the deviations from the mean position for all alpha carbons, and the blue line is for all atoms. And the important thing to note here is that if the protein is living in a harmonic well, in a parabolic energy potential, we'd expect this histogram to look like the parabola that's shown in the dotted line in between. I'm not sure if it's possible for you to see it. There's a small parabola that's actually inside both the red and blue lines. And so what this means is that, again, we're seeing deviations of the protein atoms, of its spatial coordinates, that are much more populated in these rare states than to be expected if the protein's just living in a harmonic well. So we want to find those directions which correspond to those rare extensions, because we think that they're going to allow us to access some of those substates which aren't primarily sampled by the data, going back to that scattering data plot where we saw 2 substates, but we only got 1 substate with the MD. And global anharmonicity is also very present in experimental data. This is the right histogram is for NMR data. So just to motivate exactly what that means, I'm going to show you the same movie from before, but now 4 of the atoms are highlighted with, of the alpha carbons are highlighted with yellow spheres. And the 3 other panels are the joint distributions of their spatial deviations. If I use principal component analysis, which is a second-order method, then I'll come up with, I'm essentially fitting the best fit ellipse to these distributions. And maybe that's okay for some of these distributions, but not for all. So the bottom 2 panels, it looks like the natural orientation of the data is saying that we shouldn't just look at the second-order statistics. If we use quasi-anharmonic analysis, we get the red arrows, which more naturally align with the orientation of the data, and which moreover need not be orthogonal, which is an assumption with PCA. So the next slide is just to say, how do we go about, if it comes up, finding those red arrows, but not just for 2 atoms, but for the entire protein trajectory, trying to find these directions which correspond to rarely accessed states. And it means mathematically, instead of diagonalizing the covariance matrix, we diagonalize a 4th-order tensor. And we end up with, instead of the harmonic modes, which I'm depicting here as U, we have the anharmonic modes, which I'm depicting as D. And we can now project the trajectory that we saw into that subspace and see if we can interpret the subspaces in a useful way. So here I'm showing you that exact projection for the top 3 modes, and so anharmonic modes 1, 2, and 3, and I'm coloring each conformer, which is represented as a dot, with its internal energy, the sum of van der Waals and electrostatic interactions, but without the solvent. And we were glad to see that there's this emergent property that structures of similar, let's say, internal energies seem to cluster in a spatial way. And this is surprising because the analysis that we've done does not consider energy in any sense. It's only the spatial deviations. So this gave us some confidence that perhaps this method is finding those red arrows that are actually aligning with where data sampled and where you can expect to find real you could generate real structures and perhaps even real transitions. The colors of the models on the right part of the screen correspond to the substates marked with ellipses. And what's important to see here is that ubiquitin has a highly characterized secondary structure, and most of its motions are quite subtle. So it doesn't move around as much as a big hinge protein. And in that way, if you have an analysis technique which can resolve its substates, it suggests that method is a little bit more sensitive than some others. This is now a more thorough comparison between the quasi-anharmonic modes and the standard principal component modes, which are shown in the lower triangular depiction. So these are two-dimensional projections. And you can see that this is why this method has been useful for us for identifying substates, because there's these outlier points in the QAA projections which correspond to rarely accessed states. That's what we're after. Okay, so the next part of the talk is just to say that visually we saw clusters in the three-dimensional projection. What are the clusters if we look in a higher dimensional space? That is, if we consider more of these anharmonic modes as feature vectors. And we've done that using a hierarchical clustering method where the connectivities between the different conformers, which are from this downsampled simulation trajectory, so there's a connectivity between each of the conformers, and that connectivity can actually be diffused so that you come up with a multiscale depiction of the transitions between conformers. And the transitions here indicate not an actual kinetic transition between different conformers, but just a connectivity within the QAA space, which we saw a picture of earlier. And what's nice about this is that there's no need to input, there's no user parameters for determining the number of clusters, but there's different resolution levels. So we ended up choosing a hierarchy level where we could manage the subsequent analysis, which is 78 clusters. If you'd like more detail on this clustering, there's a poster also tomorrow, Z45, that uses the same clustering for analyzing cryo-EM maps. Here's a look at the clusters if we had we sorted the previous cluster. And now I'm showing you the distribution of the memberships in each of these clusters. And the memberships of the NMR ensemble, which were not included in the analysis, but just for reference, are shown with the wedges along the x-axis. So most of the clusters, the first, let's say 20 or 30, these are arbitrarily indexed, are similar to structures in the NMR ensemble, but most of the clusters, and indeed most of the simulated conformers, are actually outside of that. And that's not surprising. We commonly see simulations that access states that aren't seen in NMR ensembles. And what's interesting about comparing this histogram with the clustering earlier is that in the higher dimensional clustering space, using the diffusive clustering, I'm showing down in the lower three scatter plots where those clusters would fall in the low dimensional space. And this is to illustrate that whereas say cluster 68 is actually very easily visualized in these low dimensional projections that we've seen with other methods, but some other clusters, for example cluster 69 or cluster 17, will be very tricky to pick out. in the low-dimensional space. And this plot is just an encouragement that whenever we're trying to analyze simulations, we shouldn't just look at the easiest visualizations of those simulations. And the next part of the talk is to explain, now that we've obtained these 78 clusters, how can we go about modeling them in a very compact way. And we've invoked an auto-regressive model, which is a common tool that's used in signal processing or image analysis. And the basis of this model is that there's some statistical signature of the data which is time invariant that repeats itself. And this is this is only possible because we've done some clustering so that we've isolated substates where we can at least hypothesize that there would be some shared, or let's say time invariant dynamics. And just as this initial, for our initial model, we've considered that each conformer, which is represented in this, again, this low-dimensional QAA space, we're calling this W, and it's related through the anharmonic modes to the actual conformer, which is X. And transition matrices in a linear way are what are actually computing the next conformer given the two previous. So it's the second order auto-regressive model. And this model is constructed at each of the 78 substates that I've identified using the hierarchical clustering. So the comparison, so just to kind of summarize where we are, the comparison between structures has been done in QAA space, the clustering has been done with our hierarchical model, and now within each of those subspaces we're trying to characterize local dynamics. And the idea here is just that we're not only interested in transitions between substates, but in trying to define in a compact way what's happening in those conformational substates. And it turns out that A1 and A2 for each of the different substates can easily be solved using some methods, pseudo-inverse and various matrix operations, so that it's not so costly. And so solving for those transition matrices is quite straightforward. And once I have them, once I've solved for A1 and A2, then I have a completely generative model of the protein dynamics, because given a W1 and a W2, W3 is now determined in this linear fashion. And I'm not showing the details of the noise because obviously protein motions is a stochastic process, but we've handled that as well in the paper. So here's a look at one such generative model from cluster 17, which was generated from almost 8,000 conformers that were projected, that were found in that cluster. And something to note about such a generated trajectory is that even though the MD itself took several months to generate the data, we can reproduce a lot of the at least superficial characteristics or basic characteristics of protein motions with these very simple models, which is, I think, quite interesting. So the previous slide gave an example of protein motion, the generated trajectory from a single substate, and now I'm going to let the trajectory move around between different clusters by simply comparing each new generated conformer frame with the cluster centers that were identified previously. And when a new cluster center is approached by the synthesized trajectory, I switch the A1 and A2 transition matrices to the corresponding ones. So just to play, once again, it's interesting to see that these motions are shown in the distribution below, where the original memberships for each cluster are shown in white, and red indicates the cluster memberships for the synthesized trajectory. Maybe it's a little hard to see, but they're in the red outline. And but one wouldn't expect them to be exactly the same, because in this case we're not modeling the kinetics in a very rigorous fashion. We're simply saying, yes, the synthesized trajectory can switch between substates. But we're working on methods for incorporating a more rigorous kinetic model of the transitions. And just to highlight some of the motions that you observed in the movie previously, now I've just colored the various domains of ubiquitin which moved the most and which also happen to correspond to those regions involved in binding. And the scatter plot on the right is indicating for the synthesized trajectory how many, what's the number of potential binding contacts with different substrates for ubiquitin. So this is to, again, highlight that the synthesized trajectory is sampling some states which we think are close to the bound states for ubiquitin. And the takeaway message for the talk, hopefully, is to stop, is just that these rare states in the tails of the distributions are important, and they shouldn't be overlooked, even though there are a lot of methods that do overlook them. And also that if one partitions the conformation space of a protein trajectory, particularly for a protein that's well folded, that there is some time invariance within those substates that can be modeled very, very efficiently. And as a result, you can generate synthetic trajectories in minutes, which are corresponding to the same amount of sampling time that's required on several months on a large machine. So this work is a collaboration between, as I mentioned, Oak Ridge National Lab, that's Arvind and Pratul, and then Chakra, my advisor, and Virginia, my lab mate, helped a lot. She has a poster tomorrow, Z45, on the clustering method. Thank you for your attention and for the funding to come.